Hip. Sorry. Yeah. 2 days already. Yeah. That's slow. Oh, I knew I shouldn't have put the butter in with the bread knife. Crumbs. Oh well, sorry about that. Well, it's been 2 days now, and your brine shrimp should have grown very well by this time. You should have been stirring it once or twice a day. That way you get air mixed into it. That way they have enough air to breathe inside, so make sure you have been stirring it once or twice a day. Now what we're going to do is, first thing we're going to do is prepare a wet mount. A wet mount is a slide that is taken from a liquid. Now to prepare your wet mount, you put a drop of water in the center of your slide. Use your forceps here, little tweezers, to very gently grab the cover slip because it's very thin glass and very fragile. Hold the cover slip at an angle and then let it fall onto the slide. This should put a very thin layer of water underneath your cover slip and trap that thin layer of water between the cover slip and the slide. Make sure you have no bubbles there, because the bubbles you'll be looking at bubbles instead of the shrimp. So make sure you have the shrimp there, make sure that there are no bubbles, and you're ready to go then. Be very careful when you hold these though, because it is glass, you can cut yourself with it. Hold it gently, and the cover slips are very, very thin. So make sure you hold those very gently when you're preparing it. Now slide your clips to the edge. Slide them back over it. Do not snap them down and crack your slide. Slide them right over the edge. If you lose one like that, just put it back on. Not a big deal. Now you're going to start with your scanning objective. Look through it. Find your shrimp at the low power. You're going to find some of the shrimp at the low power. You may have to jiggle your slide around a bit. Once you've found it, focus it. Once you've got it in focus, then move to your low power lens. Click it into place. Look in again. Focus it a little bit more to get it into sharp focus with the low power lens. Once you've got the low power lens in focus, then go for your high power lens. Now the high power lens, remember, do not adjust it much with the high power lens. You're not going to want to adjust it much. It should be in pretty close focus, but you're just going to want to move it just slightly. If you move it too much, you will crack your slide. So be very careful not to do that. So find it under there, look at it, draw it in your lab report. Now you should have printed out your lab report before this. So at the beginning, make sure you have your lab report printed. Make sure you have read all of your lab report so you know what's going on. You've read all your lab report. Refer to it as you're doing the lab to make sure you're getting all your steps in there. Also have your lab report printed out. That way you know what you need to answer. You know where your questions are. You know what you need to answer in it. You can make all your drawings there. So if you look in here, you've got to draw the sea monkeys. Look inside there. Draw them out. Now you're probably going to want to do a couple of different slides, because the brine shrimp look different at different stages of their life. So you're going to want to do a couple of different slides, see if you can catch some babies, see if you can catch some that are more mature, see what's in there. Take a look at a couple of different ones. To make it, just make another slide just like this one. Go back to the scanning lens. Take this slide off, slide the clips to the side, take this slide off, put it to the side, and then make another one with another drop of water. See if you can find one that is a different age. So that is doing your wet mount. Now, for your dry mount, that is a prepared slide. Your dry mount is a prepared slide here. This is one that has already been made. It's in this little case that's in the box of your biology stuff. Now this one says slide combo, onion root, and root nodule. You will need this slide for one of your last labs this year, so do not let anything happen to it. Make sure you're extra careful. Do not break it, because you will need this later. Now to take this one, you do this one the same way you did your wet mount, except you don't have to put a cover slip on it. You put your clips on it very carefully. Put your clips on it. Start with your scanning objective. Look through. You move the slide around a bit. Find what you're looking for. Once you've found what you're looking for, focus it with a scanning objective. Then go to the low power lens. When you get to your low power lens, focus it with your low power lens. That way you get to see what's going on there. And then you can move to your high power lens. Remember, your high power lens, do not turn the focus knob much. Just very, very slightly so that you get it into focus. Otherwise, you will crack this slide and ruin it. So make sure you are very, very careful with that. Find what you're looking for in there. Take a look at it. Draw it out also, because that's part of your lab report is to draw these out. Now, once you finish that one, put it back in its box and put it away so that you've got it again and you know where it is for your next lab. Once you've finished using the microscope, put it away, put it back on the scanning objective lens, take your lens out, put your lens back in its case, seal it back up, put your cap back on it, cover your microscope with a cloth, or if you've got the cover for it, cover it with that, or put it back in its box. Whatever you need to do to keep this safe and dust-free until your next lab. Now, something else that you can do is there's an optional part of this lab that involves some hay and water. Take your hay and your water, put it in a large container, so a large-ish container, bigger than this probably, but I mean one of these may work also. You take some room temperature distilled water again. Remember, use distilled water. Do not use the chlorinated water you get straight from the tap. Buy gallon distilled water or bottles of distilled water or spring water or whatever. Take your hay, put it in your container, pour the water in with it, let it sit for about a week. Now you're just going to let it sit in a place that has some sunlight, not a lot of sunlight because you don't want a lot of UV rays, but you want it to be lit, you want it to be warm. Let the stuff grow. It'll probably start smelling a bit. It'll start getting kind of murky. That's fine. You've got stuff growing in there, and that's the thing that you want to do. Once you've got stuff growing in there, do the same thing you did for your wet mount. Take a drop of water out, put it on a slide, prepare the slide, look and see what you can find in there, because you can probably find all kinds of little critters in there. And if you do that part of the lab, talk to your teacher about extra credit for it. I can't guarantee that because that's up to each individual teacher. But it's a possibility you might be able to get extra credit for doing that second part of the lab with the hay and the water. Remember that once you have completed your lab, clean all your stuff. So take your slide cover off, clean that with a soft cloth, very carefully because these are very fragile. Clean your slide with a soft cloth, put both of them back away where they're supposed to go. I will do that once I have cleaned them. And you're ready to go. Well, I hope you've enjoyed this lab today. If you have any questions, please make sure you ask your teacher. This lab, each and every lab that you do is worth a big portion of your grade. So they're not very difficult. As I've shown you here, you've got all the instructions. Just do your lab, get it turned in, get your points. Don't end up making a lower grade than you could just because you didn't do your lab. These are a fun part of the total science experience. Well, up until your next lab, hope you have fun and be safe. Bye, everybody.